Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello, everybody. Welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the OCR A-Level Biology A Paper 2 from June 2017. Membranes are found within and surrounding cells. Which of these statements A to D is not a role of membranes in cells? So access vary between areas. That is true. It does. It compartmentalizes different bits. Cell signaling is indeed correct because they have receptor proteins on the cell surface membrane, so that's out. Provide support for the cell, no, that's not true. Their entire role is to compartmentalize and separate things out. And then site for chemical reactions, well, there are some areas where chemical reactions do happen, like the intermembrane space of the mitochondria, so that one's out, so it's got to be C. So which of the statements about amylopectin is correct? So one of them is that it has 1,4 and 1,6 glycosidic bonds between alpha glucose monomers, so that is indeed correct. It's unbranched, it's not. Amylose is unbranched. Amylopectin is branched, so that one's out. It has 1,4 and alpha 1,4 and beta 1,6. It doesn't have beta in it because beta glucose is only in cellulose, and D as well. Beta glucose is only in cellulose, so it's A. So this shows a blood smear, which correctly lists the cells that are visible within the smear. So I can see immediately we've got these multi-lobed nuclei. So that means immediately neutrophils are present. So we can immediately take C out. Next, I can't see any cells where there's a big nucleus like this, a singular one, which is a lymphocyte. So that means I can cross out the ones that have lymphocytes in them. So that's D, right? So now we're stuck between A and B. Erythrocytes are red blood cells, which are indeed here. There's no lymphocytes in both. That's fine. But then there are neutrophils in both. We need to think about monocytes. So monocytes, remember, have these big fat nuclei that are just like this, and it kind of looks like a kidney bean almost. And there's none of those there. So monocytes are out, so it's B. The haploid number of this species of koala is 8. Independent assortment of chromosomes and meiosis contributes to genetic variation in the gametes of the koala. How many genetically different versions of koala gamete would be possible for one individual to produce if independent assortment was the only source of genetic variation? So when we're thinking about independent assortment of chromosomes and meiosis, what we're thinking about first is independent assortment of homologous chromosomes, which is like maternal, and then paternal, right? So these are not homologous. These are just double, copies of each homologous pair. In meiosis one, the homologous pair split, and then here, the paternal one split here as well. Now it's asking though, the haploid number in this is 8, right? So its diploid number is 16, right? And it's saying how many genetically different versions of koala gamete could be produced. So our options is, imagine we've got a singular gamete cell here. We need to put haploid, right? So we need 8 different things. So 1, 2, 3, 4, 5, 6, 7, 8, right? We have one of two choices, maternal or paternal. So that means we've got two choices here. The next one is we also have maternal or paternal, times 2, times 2, times 2, and so forth. So the number of combinations you can have is 2 times itself to however many times your haploid number is. So here it's 8, so we're going to have 2 to the 8 different possibilities, which is 256, which is C. Which of the following processes occur during DNA replication? So with the whole point of DNA replication is you've got two DNA strands that are complementary base paired with each other. I'm going to separate them. You have each one act as a template. Nucleotides line up next to them like this using complementary base pairing. And then DNA polymerase joins them together with condensation reactions to make phosphodiester bonds between adjacent nucleotides. Not this way, this way. So then we need to figure out which one that is basically. So number one is that breakage and reformation of phosphodiester bonds? Nope, because that would break the nucleotides. That would break the strands into two chunks, which is definitely not happening. Number two is breakage and reformation of hydrogen bonds. That is indeed correct because they're separating the two strands. So that's the one. And then alignment of free nucleotides at their complementary bases. Yes, because that's how they actually get there. And then phosphodiester bonds are only formed, not broken. So in that case, 5 has to be C, only 2 and 3. So which of the statements best define species evenness? So species richness is the number of different species you have in an area, right? So like, I don't know, this type of squirrel, this type of, rabbit, this type of tree, whatever. Species evenness is a measurement of the relative abundance of each species, as in how they're actually how many numbers of them there are of each species, right? So whichever one of these is going to be that. So the number of species in an area isn't definitely not it. That's species richness. The relative abundance is, well, whoa, would you look at that? That's what I said. The relative number of species of the number of individuals of a species, that's a population, and the spread of species over an area is their distribution. So here, the relative abundance is going to be B. The first stage of primary succession is the pioneer community. Which of the following statements about pioneer community are correct? So here then, in primary succession, what you have is essentially you've got this really, really barren land, like a rock or, like something just not very nice, like very sun exposed dry area. A pioneer community can live in this and it's happy and it's chilling and whatever. And then eventually they die and they make up a little soil. And over time, more things come in and die and this and that, and blah, 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 until you get loads of extra things happening and all this stuff, right? We're going to zoom in a little bit on what the pioneer community are doing because that's what this question is related to. So the pioneer species are able to survive in these harsher conditions because they have adaptations that let them do this. The adaptations that they have is that they're able to tolerate extreme environments, like for example, really hot. They also have seeds that germinate rapidly. They're able to photosynthesize themselves using light and rainfall and air and CO2. And they're able to fix nitrogen from the atmosphere because if you think about it, it's just rock. All they have is CO2 in the air, atmospheric nitrogen, and sunlight. And as well as that, they're also able to make large numbers of seeds and spores, which can blow around this new area and allow them to, colonize that area. So if we have a look then, the options are species produce large numbers of wind-carried seeds or spores, which is indeed correct. Their biomass is low, that's correct, because there's not very much of them. There's just a teeny amount, sort of growing and starting. Like, for example, lichen, they're not very, biomass heavy. And then many species of lichens and mosses, well, yeah, that's what they tend to be a lot of the time, so it's got to be A. The graph shows a typical population growth curve, and it says which row describes what is happening at each of the stages V to Z. So if we're going up no matter what in population size over time, it means that there's more births than deaths. So any one of those where death is higher is going to be knocked out whenever it's going up, really. So here, the reproduction rate is higher than the death rate because it's starting to climb up, no matter what. At W, what's happening as the time goes on, the population is started to increase a bit more fast, and then here it's rapidly increasing. Y, it's increasing less steadily, and then Z, it starts to level off, and the population becomes constant, as you can see here. So whichever one of these fits the bill is going to be it. Now, if we just look at this here, this column, all of them are the same, so there's nothing that helps us differentiate it here. As time doubles, population doubles. As time doubles, population more than doubles, and as time doubles, population doubles, and the population is increasing rapidly, right? So let's just have a think. If I take, for example, here and here, and then I just move another chunk like there, you see that this has gone up more than a double would have been. And the reason is because this is exponential growth. So if it was a linear growth, it would just be this doubles, right? Where it's just going one straight line. When it's exponential, it starts off and shoots up. So immediately you're out. This is kind of the same. So we're stuck between B and D. So next, if we look at X, here reproduction rate is much higher than death rate. That is true. Otherwise, it would just kind of be like here. Here, it's so much higher than death. Whereas reproduction rate is much higher than death rate. That doesn't help either. So now we're still looking at B and D. Population growth is slowing. Population is decreasing. There you go. That's the one that's out. Because population growth is slowing is what's happening at Y. Not that it's decreasing. It's still going up. If it was decreasing, it would be doing this. It would be going down. And that's because the population size, remember? Like, you can see it's going up. So that one's out. So it's B. Now, cut pieces of agar jelly can be used to investigate the factors affecting diffusion rates in cells. 4 pieces of agar jelly containing a universal indicator was soaked in the same concentration of hydrochloric acid for a minute and then they were blotted dry. Which of the following pieces of agar jelly would be the first to turn entirely red? So it's the one with the smallest edges, really. That's like a nice little teeny piece. So we want to maximize the surface area and not have too high of a volume. So for example, if I had this really thick cube here, like this, or cuboid really, right? And it was a piece of ice and I gave it to you, it takes some time to melt. Whereas if I gave you this thing, the same sort of face side here, but it was really thin instead, like that, then it would melt a lot quicker. So ideally, we want some sort of cuboidal shape like this, where it's, very high surface area and very little volume. So if we have a look with a cube of 4 sides each, right, that's going to be like a pretty nice just cube, which is going to take a bit of a while compared to the others. So maybe not you. A cuboid with edges 2, 4, and 6, right? So 2 by 4, and then 6. That looks pretty flat, right? So that looks like it would melt. It's like this on its side, so it's looking tempting. 3 by 3 by 5 would be this. So again, it doesn't have as much of a higher surface area as this does. So that one's out. And a sphere with diameter 4 is going to have a lot less than this because it's just a sheet here. So in that case, it's got to be B. So which of the following options, A to D, is a primary defense against pathogens? So what we mean by this is we're looking for non-specific defenses that are trying to stop pathogens getting in. So the things that we have is, first of all, we have our skin barrier, which, physically stops them. And it also makes things like, it has like flora on it, and it also has a sebum, which is an oily substance that inhibits the growth of these pathogenic things. We have mucous membranes like in our eyes and our mouth and stuff, and, other places that makes like mucus, which traps stuff as well. We also have lysozymes in our tears and urine. So these are all things that sort of non-specific, again, that just try to stop things getting in or obliterate them, just generally speaking. Another thing that we have, though, is inflammation. The inflammation response is where you've got heat, redness, swelling at something that's become inflamed. And the reason that happens is because you have mast cells which release histamines and cytokines. So the histamines, what they do is they make your blood vessels dilate, right? And they also make them more leaky, so the cells can actually get out into the affected tissue. And cytokines attract phagocytes to affect the site, the site that's area. Cytokine means cyto, cell, kine movement, cell movement, right? So the moment the phagocytes kind of come into the picture and stuff, that's more of a specific thing because they're starting to go for a specific pathogen. But here, these are all just things that happen in a general sense. So looking for anyone that is that. So antibody production, no, because that's specific. You'd have to, stimulate the right sort of B cells eventually to do that. Inflammation is again non-specific because even if I smack my hand against the table, I might get some inflammation going. Phagocytosis, no, because that's kind of again more specific. It's coming into this process when it's starting to recognize that, oh, this is what it is and stuff like that. And the cytokine's been released. And the same for T killer cells as well. It requires a specific, interaction between, like, for example, an antigen presenting cell and a helper T cell and things like that. So immediately it's B. The diagram below shows a plasma membrane. Which of the following labels point to a structure that contains a sulfur atom? So to me, a sulfur atom immediately rings bells for disulfide bridges in proteins, right? So whichever one of these is going to have that. So 1, that's a protein. 2, this is a glycolipid. And then 3, that's your phosphate head from your phospholipid bilayer. So the only one it can be is D, because that's a protein. Which of the following statements about gene therapy is not correct? So changes resulting from gene therapy cannot be passed into offspring. Well, I mean, if it's germline therapy, it can be. So that's actually it. It's just A. But if we have a look, germline gene therapy affects the whole organism. That is true. Gene therapy is a form of genetic engineering. That is true because you're changing the base sequence of things. And then somatic cell therapy can only affect a limited number of cells. That is true. Germs in biology means gametes. So if you're affecting the gametes, every single cell in the offspring is going to have that. And somatic means body. Soma, body cells. So body cells. So it means only going to do it in body cells that aren't gametes. Below are 3 processes that occur in living organisms. Which one of these is important in determining the body plan of an organism? So in a body plan, what you need to do is you've got to do cell division, mitosis, to actually create the right cells, but you also have to self-destruct cells that you don't need anymore, which is going to be apoptosis, programmed cell death. So that one's that one's that. Meiosis, the only existence for it is to make gametes. So that one's out because we're not going to use that to make a body plan, so it's B. 3 methods of pathogen transmission between animals and plants are listed below. Which of the following methods of pathogen transmission does not happen to plants? We need to think about how these actually, what these describe. So we're talking about communicable diseases and how they're transmitted, right? So as in how basically we are spreading disease. So you can do direct transmission, which is where the pathogen is directly transferred from one person to another, or one individual to another, through things like direct contact, so skin-to-skin contact, kissing, sexual intercourse, things like that. Inoculation, so you break the skin and something gets in, or an animal bite. Ingestion is eating as well. Indirect is where the pathogen travels from one individual to another through an indirect means, so like fomites, for example, like bedding and cosmetics and stuff like that, right? Or droplet infection through inhaling something, like for example, if somebody coughs or sneezes, or a vector as well. So, mosquitoes that transmit malaria. And also water as well can be itself the vector. Now, for plants, it's a bit different because plants don't, cough, sneeze, shag, things like that, right? But we still get plants spreading through plant communities. So when you've got direct transmission in plants, what happens is you've got direct contact of a healthy plant with any other part of a disease plant. So for example, tobacco mosaic virus or something like that. Indirect transmission tends to be the sort of similar principle, but in the context of plants. So soil contamination, or vectors in the wind, water, or animals like carrying them and when they're scrying them, or like birds carrying them, or bees, whatever. Or humans as well doing that. Direct contact is one of them because if it grows, onto like a plant that's affected, that's true. Vectors is also another one because again, like, you've got wind, water, bacteria, all those sort of things that I've just said. So that's another one. And 3 is droplets. So again, plants don't really inhale, so there's not really going to be any of that going on. So it's just these 2, B. So here's a European badger, and it's a member of this. The American badger belongs to a different genus within the same family. Which of these options is the correct binomial name for this American badger? So it's not expecting you to know what this American badger is. And how do people bring this to me? And it had me baffled for a long time until I realized exactly what it was, and it was very cheeky. When you're writing a binomial name for an organism, it's always its genus in italics and then its species. But the genus is always capitalized, and that's in lowercase. Here, it's told you that it's in a different genus, which is Meles and Meles, so that one's out. Here, this is lowercase, so that one's out. Here, this one's lowercase, and that's uppercase, so that one's out. The only one it can be is D, which is just cheeky as hell, but just be careful. So the Madidi National Park in South American rainforest is a home to a wide variety of species. The largest predator in the individual is a jaguar. These are well camouflaged and hunt mostly at night, and a single individual can cover large areas. In 2007, the Wildlife Conservation Society attempted to estimate the population. Digital camera traps were placed in areas that they would likely visit, and if an infrared beam was broken by a animal, then the camera is activated, and the camera then took a picture. So just why it's not appropriate to estimate the number of jaguars using the capture recapture technique. Imagine if you had to capture a bloody jaguar and then, like, I don't know, put some like paint on it and then just like send it back into wild. Like, come on, let's be real here. You're not doing that. So number 1, immediately, that is dangerous for both the jaguar and the collector. And number 2 is there anything else in the question that's hinted at you? Well, here, we also need to think about the factors that could affect your mark release recapture stuff, right? So if I have a very low number of them, imagine I only have like 1, 2, 3, and I take one out, we've now gone from 3 to 2 out of 3, which is 33% less, which looks like, a lot and has a bigger impact, right? Whereas if I have 1, 2, 3, 4, like 100 of them, right, and I've taken 20 out, then we've only lost 20%. So can you see how a bigger number of them to begin with has a less of an impact? Even though I know you're putting them back in, it's just not going to give them it's going to screw up the method. You need a really large sample size, generally speaking, for this kind of stuff in whenever you're measuring anything. So here, this is going to give a pretty bad estimate because you've only got low numbers. And the reason is because, like, again, with those three, right, I've taken that out. One of them's now 33% of all that stuff, right? But then if I put this marked one back in, right, then when I count my second sample, let's say I've got two, then I'm going to pick up two of them again. I'll be like, oh my God, wow. Like, oh my God, the sample size is, bigger or smaller, whatever. Like, it's just not very reliable. So here, the idea is that there are low numbers, so the estimate's unreliable, or inaccurate, really, should say. It's not going to be close to real value. So most studies estimate the population density to be 5 individuals per 100 kilometers squared. In a 2007 study, 100 cameras were set up covering this area and 28 images of 9 different jaguars were recorded. How well do these support a population estimate of 5 individuals per 100 kilometers squared? So we need to do a bit of number crunching. So if they've told you that they're estimating 5 per 100 kilometers squared, we have 100 cameras that cover this area and 28 images of 9 different jaguars were recorded, right? So we have a look. These images eventually culminated in 9 jaguars for 271 kilometers squared. So that means 1 kilometer squared would have been 9 over 271 jaguars. So then 100 kilometers squared, which is what they've said, per 100 kilometers squared, would be 9 over 271 times 100, which is roughly 3.3, or 3 jaguars per 100 kilometers squared, right? So this already tells you that this is lower than an estimate. So you would say that this 3 per 100 kilometers squared is less than the estimate. Therefore, it doesn't support. It doesn't support it, basically. We need to think about possible reasons why as well. So if you look at the reasons why, it could be that, for example, we have low reliability here because we've got 28 images of 9 different jaguars, right? That's not very helpful. Like, we don't know if it's been repeated. We don't know how many of them are. We've only got 100 camera traps. So there's like, no information about no info about repeats and stuff like that, for example. So therefore it's got low reliability. And we don't know about its reproducibility. The other thing, though, is when it says something like, oh, do the things support this and that, blah, 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 how well? It's, again, a kind of like almost disguised evaluative type question. So this is a contradicting statement. What about supportive? Well, in support of it, I guess you could say that 3 is close to 5. So therefore there is some support, right? And then, again, another reason might be that this has gone wrong is that some individuals might not have been captured, is what I would say, by the cameras. They put 100 cameras in an area that big. That is huge. So they might have just missed some of the jaguars. And also, they only got 28 images of 9 different jaguars anyway. That's also, like, not very helpful. And also, they're multiple pictures of the same jaguar. So, like, if we didn't sort of have that going on, like we had a bigger area, for example, more cameras, we'd probably get some better results. Other evidence used to estimate the jaguar population includes footprints and reports of sightings by local humans, suggest one disadvantage for each of these. So, I mean, one of them, first off the bat, is that, for example, human sightings, you might, like, misidentify them. You might not be able to tell what it is. You might be like, oh my God, it's a jaguar. When in fact, it's like, I don't know, some of the weird big feline. As well as that, it depends on memory as well. So people might forget. So it depends on memory, so may forget. It could be that people exaggerate or lie. And also, as well as that, like, let's be real here. Like, you're going to, unless you're going on like a tour or something, are you going to be around jaguars very much? Probably not. So you're not going to be, it's not going to be very likely you're going to be in a place where there's, places to be and see jaguars. Footprints, the problem with footprints is, again, maybe they misidentify it, or it might even disappear over time, because obviously, footprints, like, are hard to look at. They might, like, get stood on and trampled. So misidentification. Or, remember, these are just one. Don't write 2, please. Just write 1, but I'm only just giving it as an example. You could have misidentification, or it could be that the footprints disappear. And also, like, I mean, if you've seen, like, the Bigfoot stuff, dude, like, I'm not... So the Medina National Park is also home to approximately 260,000 humans who support themselves by means of cattle farming and the production of timber and Brazil nuts, a large nut harvested from a local native tree. Conservationists have been working with local people to promote sustainable uses of these resources and government agencies to maintain the quality of the national park. Explain why this is an example of conservation rather than preservation. So, conservation is where you're protecting an ecosystem and managing it actively so that the natural resources in them are sustainable and they can be used without running out, right? And it's a process that involves adapting your approach and changing it depending on what the ecosystem happens, what's happening in the ecosystem, right? You're actively going in there and doing things yourself, whereas preservation is different. It's where you're protecting an ecosystem to keep it exactly the way it is. You're not taking things out, you're not doing anything that could damage them, and you're limiting them. For example, like the Antarctica, right? Like we can't go there to like mine or do anything too heavy there, and there's very limited tourism and stuff because we want to keep it the way it is. So here in this question, they've told you that they're working together to promote sustainable use of these resources here and also to maintain the quality of this park. So they're actively doing more things here. So number one is that the reason it's conservational or conservation, sorry, is because there's actively, people there. So conservation because people are there. If it was preservation, we want to keep it as is so people wouldn't be there. And also because the area is used for logging and also like Brazil nuts. And lastly, the main key point is that there are active measures or management steps to maintain the park. That's what sets aside conservation versus preservation. So the sweet pea plant has been used to study inheritance since the 19th century. The seeds of the sweet pea plant can vary in color and shape. The gene that controls color has two alleles. So people use the word gene and allele interchangeably. A gene is not the same as an allele. An allele is a different version of the same gene. So for example, if you're blonde, you have blonde hair, I have black hair. We both have the hair color gene. Yours is blonde, mine is black. It's not that we're changing genes. If you change genes, you're not talking about the same thing no more. So here, what I always like to do is I like to just scribble down what's going on because if I don't, I get lost. So here it's dominant, capital Y, and it's yellow. And then little y is recessive and it's green. And then here it's the shape of alleles, and big R is round, little r is wrinkled. So these are two separate genes. In the 19th century, Gregor Mendel crossed a pea plant that was heterozygous for both seed color and shape with a pea plant that had green and wrinkled seeds. Lists the gametes that would be produced by a sweet pea plant that was heterozygous for both seed and shape. So if we have a look at what was heterozygous for both seed and shape, it would have been big Y, big R, little r. Now, people get confused on this, and I don't really get why, because here we go. Look, imagine I've got a diploid organism of CC, right? This is a monohybrid cross. I'm only dealing with one gene. Now, gametes are haploid, remember? So if you have haploid gametes, you're going to split this in half because this is diploid, one copy from each, right? So if you're making the gametes, it'd be haploid, one copy of each of these things. That's what that is, right? Everyone's pretty okay with this one. Here, this is a dihybrid cross because we're doing two different genes, one for the color, one for the shape. It's literally just this, but combined into one, essentially. So what I mean by that is that this is diploid. We've got two copies of each thing, one Y, and one R, one R. So the gametes are going to have one copy of each thing. So it's going to have one Y and one R of some sort. And that's what it is. So one Y, one R. And then it's going to be all the other possibilities. It could be little r, little y, and big R. Or it could be big R and it could be, little y and little r as well. Or it could be, I feel like I'm missing one. Why am I missing one? Oh, big R and big Y and little r, like that. That's what it all is. It's literally just that. So here then, the gametes would be what I've just written there, to be honest. So big R, big Y, big R, bigger Y, little r, little r, Y, big R, little r, oh my god, little y, little r. Then it says, list the genotypes of the offspring that were produced from Mendel's cross and then state what their corresponding phenotypes are. So what the geezer crossed was, where are we? A heterozygous for both seed color and shape, and with a pea plant that had green and wrinkled seeds. So remember that green and wrinkled, you had to be recessive for both. So little y, little r, little r, right? If we cross those, then if I just do a thing here, the gametes that would have been produced from the one parent would be here. So big Y, big R, big Y, little r, little y, big R, little y, little r. And then for the other wrinkled thingamabob, all the gametes would just be, remember this one Y with an R, one Y with an R, and all the possibilities of that. So it would just be little y, little r, little y, little r, little y, little r, little y, little r, right? Now, when you're doing these crosses, you need to make sure that you keep them consistent. So I always stick to doing, and I think it's best to, dominant first, then recessive for the other thing, then dominant first, and then recessive for the other thing as well, like that. So in this sense, what I mean is we're going to have big Y, little r. We're going to have big Y, little y, big R, little r. We're going to have big Y, little y, little r, little r. We're going to have little y, big R, little r. And then little y, little y, little r, little r. And then we're going to have big Y, little y, big R, little r, big Y, little y, So after doing all of that, your genotypes are going to be big Y, big R, little r, big Y, small r, small r, small y, big R, small r, small y, small r, small r. Then it says the phenotypes. So this is yellow and round. This is yellow and wrinkled. This is green and round. And this is green and wrinkled. So when Mendel crossed two pea plants that were heterozygous for both seed color and shape, the ratio in the offspring was 9:3:1, which is what we love. Students tried to recreate the investigation using a modern variety of pea plant that showed the same phenotypic variation. So what they did is they crossed the modern plants that are heterozygous for both seed color and shape. So where they've done two heterozygous, right? So that's going to be this times also crossed with this. Okay, that was what you would expect to give you your 9:3:1. The results of this cross were this. So it says here, they used the chi-square test to compare their data to the expected 9:3:1 ratio. Use this formula and the table for working out. So when you're doing the chi-square test, you are going to work out the observed, your expected, and then you're going to work out the observed minus the expected, observed minus expected, and then you would square it and then divide it by the expected like that. Just, you can do it in steps if you want, but I'm just going to do it this way. So if we have a look, what we observe is what you see. So 58, 31, 21, 2. Now, if it's a 9:3:1 ratio, we're expecting to have all of these split up in 9:3:1. So what we do is we work out this sigma, which is just a fancy way of saying the total because I'm lazy at writing total. 58 plus 31 plus 21 plus 2, right? Which is going to be 112. The total number of them. Now, if we want to split them into a 9:3:1 ratio, then 9 plus 3 plus 3 plus 1 is 16 shares in total. So if you do 112 divided by 16, one share is 7. Okay? So if it's a 9, one share is 7. Remember, it's a 9 times 7, that's 63. That's going to be 21, 21, and then 7 like that. So that's what we're expecting to see. So our observed is 58, 31, 21, 2, and so forth. Here it's 63, 21, 21, and 7. So then to work out the observed minus the expected squared, it would just be, for example, here, 58 divided by, minus 63, all squared over the expected, which is 63. Now, if I just skip ahead and do that, we eventually end up with 0.40, 4.76, 0, and then 3.57. Now, this formula, chi-squared, this thing means sigma, which is sigma. It's adding up all of these, right? So if you're doing sigma of the observed minus the expected squared over e, it's basically all of these added up. So if you do that, sigma observed minus e, this, all of this, blah, blah, blah. You can add all these up, which gives you 8.73, like that. There you go. Now here it shows you a probability table, and it says, after analyzing the results, the student stated that the inheritance in seed color investigation was different from that in Mendel's. It says, using table 17, discuss whether these support the statement. So number one, what we need to do is we need to figure out what we use from this table. So this table tells you the critical values for p and whether it's significant or not with every p-value. So the chi-squared test, what that's doing is it's testing the p-value is. It's looking at whether the difference between the observed and the expected is statistically significant, as in, is there a less than 5% chance that it's probability that's due to chance? What we've done is we've just worked out 8.73, right? That's our value, and we're going to compare it to a critical value here. We need to work out the number of degrees of freedom. The number of degrees of freedom for a chi-squared test is your number of classes minus 1, as in your phenotypes minus 1. So in this case, our phenotypes are 1, 2, 3, 4, so we've actually got 3 degrees of freedom. So it's going to be 4 minus 1, which is 3. So what we do is, can you see that column says degrees of freedom? We're looking at this entire row here. Now the p-value that we want to look for, the level, is going to be 0.05, which is 5%. So what you do is you go down to wherever it is, degrees of freedom of 3 and p of 5, and you can see that the value here is 7.82. What that means is if your chi-squared value is bigger than this, then it means that the p-value is less than this, if that makes sense. So if we worked out that it was 8.73, then can you see that's higher than that? Our p-value for testing whether it's due to chance or not is actually lower than that. Therefore, these results, as in the difference between the observed and expected, is actually significantly different. So that's what you would say. Is number one, you need to justify what you're about to say. So number one is that our critical value, write CV for short, you write the whole thing obviously, is greater than 7.82. Therefore, the probability of the difference, obviously write the whole thing, between observed and expected being due to chance, because that's what the p-value is testing. Whenever you're doing a question on this, what is the p-value testing? So t-test, the p-value is looking for the difference between the means. The p-value for correlation is to look for the correlation between two things. The p-value here is the difference between observed and expected. Can't just say the results. That's not helpful. So the difference between the observed and expected being due to chance is less than 0.05. Therefore, the difference is significant. A ratio that's different from this expected and across such as this can be the result of epistasis. So just explain one reason other than epistasis why the phenotype might not be this. So epistasis, just a side note, epi means after, stasis means stop, after stoppage, right? So imagine that here, this is you very beautiful bloke, or blokette, or bloke them. Shout out my NB people. Here's your lovely hair of whatever, right? You have black hair in this picture. If you are bald, which no hate to bald people, it doesn't matter if you have a black hair allele, you are bald. So if you have the black hair allele, but you got no hair, no matter the no hair is masking the expression of the black hair allele. That's epistasis. So we need to think of other reasons why there's going to be differences in your expected 9331 ratio. Another reason is called autosomal linkage. Autosomal linkage is where, first of all, it's an autosome where they're on the same chromosome that is not a sex chromosome. And what happens is that, for example, here during crossing over and all of that jazz, right? If they're very close together, the two genes that we're looking at, like for example here and here, they're not going to get crossed over, so they get passed on eventually together like that. So the seed color and the wrinkle like that. If they get passed on together, then it's more likely the gametes that end up being made are going to have more of these. For example, if the parent had big R and big Y, they're going to make more big R, big Ys than the other gametes, if that makes sense. So then what happens is you end up with a higher proportion of gametes that match the original parent if they're linked, because they're close together, they're passed together, and not actually exchanged. So that's what we're going to say. So our suggestion here then is that there's possibly going to be autosomal linkage, and it could be, well, the reason is because of both alleles on the same chromosome. So what ends up happening is you don't get any, no crossing over and independent segregation or assortment. So then what happens is both the alleles end up in the same gamete. So then you get a higher likelihood of those ones being made, which means it's screwing up. So whenever you have something that isn't a 9331 ratio, it's either epistasis or autosomal or sex linkage. Here, it's not really going to be sex linkage because we're talking about plants here, and there isn't really an XY chromosome in this context. So no. The yellow color in pea plants is the result of an enzyme that breaks down chlorophyll, which is green. So chlorophyll is green, it gets broken down, and it becomes yellow. The big Y allele codes for the production of an enzyme that breaks down chlorophyll. The little y allele is a result of a mutation in the big Y allele, and the little I forms an inactive form. So let's just really quick work out what's going on here. So chlorophyll is green, and it becomes yellow if you have big Y, right? That's what it's told you. Whereas if you have little y, then you don't have this happen. Cool. Then it says, outline how the Y allele codes for the production of this enzyme, and then explain why the Y allele codes for an enzyme with a different primary structure. This should be 3 marks. Whenever you want to make a protein in the entirety of the universe, you need to use your DNA at some point. The DNA base sequence, the triplets tell you what amino acid you make. Eventually that gets transcribed and translated, and then you end up getting your amino acids being put together at the ribosomes, and then you end up getting your proteins, your primary, secondary, tertiary structure. The amount of times I'll ask people oh, what do you think's happened when this protein's changed shape? And they're like, primary structure's changed. I'm like, right, okay. So then what's also changed? Secondary. And then I'm like, okay, tertiary. And I'm like, yeah, fine. Anything else? Any other reason why all of a sudden this amino acid sequence has changed? And they're like, I don't know. It's because the dang DNA base sequence, man. It's all about that. That's what ends up instructing how this is going to go, how it's going to go. So you would write that, first of all, as how we actually, this allele codes for it is number one, is that DNA based triplets determine the amino acid sequence, right? That's number one. Then what happens is you get transcription. This DNA is transcribed into mRNA via RNA polymerase. What happens is the exposed region of DNA, so we're breaking up the strand temporarily, region of DNA happens, and you've got RNA nucleotides line up via complementary base pairs, and then RNA polymerase joins them together by condensation reactions to make this, to make the mRNA. RNA polymerase joins them together, and then you end up getting mRNA, which, you can say as well if you want it spliced and this and that, right? But at least we've got mRNA. That's transcription. Then what happens is translation. The entire point of making a protein is you've got transcription and then translation happens. So the mRNA arrives at a ribosome, tRNA with a complementary anticodon, which is just a triplet. Obviously, you would define all these terms to the mRNA, brings amino acid, then another, more specific amino acid, sorry, then another. They're joined via condensation, makes a peptide bond. Then eventually it makes the primary sequence of a polypeptide. So then eventually you got your polypeptide, right? But then it says in the next part, it says, where is it? Explain why this Y allele codes for an enzyme with a different primary structure. So that's what I was saying before, is the only thing that determines this primary structure, well, eventually the polypeptide and stuff as well, is going to be the amino acid sequence. So what happens is there could be, so for example, a mutation of some sort. So it could be, so mutation just means a change in the DNA sequence. Eventually that could lead to something like a substitution or something like a frame shift. So then you have a different DNA triplet, which I should have said codon before, whoops. So then you get different codon, different, different tRNA, anticodon, different amino acid, different primary structure, and so forth. Loads of people just cannot put the pieces together for some reason. I don't get why. Now it says, with reference to the proteins coded for by the seed color gene, explain why this is recessive. So if we have a look, the only reason it's recessive is because you would need two little y alleles to, synthesize the non-functional version. Doesn't matter if you have a mutation. If the other one's dominant, then you're chilling. So the potato plant is a staple food in many parts of the world, and potatoes are susceptible to infection by a pathogen which causes a disease known as potato late blight. The most visible sign of this is brown discoloration of the leaves. Some varieties of potatoes are resistant to infection, suggest two ways in which an individual plant could respond to an infection. So number one is, it can make callose, which is a physical barrier. Callose. What happens is this is it acts as let me write this down, sorry. So physical barrier. What it is it's a polysaccharide, and what it does is it deposits between the plant cell wall and a membrane in times of stress. So here's your cell wall between the plasma membrane, there's callose there, and it prevents the pathogen from being able to actually, get in. And that's quite helpful. Another thing as well is they can make and produce and release toxic chemicals to inhibit growth, right? So you can't really say like antibacterial insecticide and stuff like that. It's not really, first of all, insecticide, not really, like that's kind of like, what we use and we make and pheromones know. What we're trying to get at is what this thing can do to respond to an infection, and it releases toxic chemicals, which will kill that thing or cause harm to it directly, if that makes sense. Other things as well is it could die, the tissue could just necrose, and also leaf drop as well. It can just get rid of the leaves. It's ways of just trying to stop the infection getting in. The resistance of different varieties of this kit was investigated. 3 different clones, A, B, and C, were used. The clones were grown in different adjacent fields at the same time period. The percentage of leaf area affected was estimated. So let's have a look at what their dependent and independent is. That's always helpful. So independent is the actual, where are we going on here? We've got the clone, whichever one it is, A, B, and C. And what's changing then is, well, so that's that. Sorry, and then what we're measuring is the percentage area of a leaf with an infection. Right. Next, what we do is I have a look and see what the patterns are. So, for example, this is so hard to read. Oh my god. So that's clone A, C, and that's B. There we go. So if we have a look at A, number 1, compare one by one, then with each other, and then everything else, right? So time after planet days, area with infection, clone A had a not great time. It was kind of going up, and then shoots up, and it actually had the most at the end. In fact, nearly all of the area of leaf was infected. Here then, for B, it started going up and up and up, and then it kind of leveled off and stopped at this point here, or stopping, right? And then C could put up a decent fight before it started going up. Fine. And then between all of them, A had the most infection at the end, C did, and then B. Maybe this was useful. We'll have a look. So here it says, suggest why it's important to use clones in an investigation such as this. Whenever it's asking you about why is it important to do X and Y, you need to be thinking what can screw up the dependent variable, which is what we're measuring. As in, what's going to be needed, what do you need to control? So here the entire point is that we're going to reduce genetic variability, because you don't want like different ones having different, capabilities and adaptations, which is it's a controlled variable, which then increases the validity of this experiment. It's not really a valid experiment. For example, if I go and give 10 people in a room who I have no idea could be allergic to chocolate, give them chocolate, they all shit themselves, and then say, yeah, they shit themselves, because I've not controlled for the fact that they might be allergic, so it's not valid. Now it says, state how a clone of potatoes can be produced in this investigation, explain why it's important to do this under aseptic conditions. So one thing you can do is you can do tissue culture or micropropagation. So I'm going to go with tissue culture. And then it says asepsis is important because we want to reduce contamination. That's it. That's the whole point. The extent of infection is estimated by comparing the area under the curve of the graph. The area under the curve of this clone B is on 250. The units can be ignored. Calculate approximate area under the curve between day 135 and day 98 for C. All we're going to do is we're looking at C, so we're going to split it here, and then we're going to make another parallelogram here, or you can make another triangle if you wanted, for example, whatever. And yeah, find the area of this by doing, the height here times this. You could do the parallelogram, which is half a plus bh, or you could do another triangle and find the area here, and the area of a triangle, which is half abh. That's all you're doing. Just for the sake of time, I'm just going to let you guys do that, and you end up getting, where would you look at that? I'm so fast and I'm so good. 952. Then it says calculate the area under the curve of C as a proportion of the area under clone B. So as a proportion means as a fraction. That's another way of saying it. So for example, that means it's going to be C over B. So in this case, it's going to be 952 over whatever B's area was, which they told you was 1250. If we do that, we get 0.7616. There you go. So then we stick to the number of significant figures, which is going to be 0.76. Using figure 18, suggest why the area under the curve is used as a measure of infection rather than the area of the leaf that is visibly affected on a given day. Because if we have a look, this is the area under the curve, and we're looking at it over time. So what we're seeing is that the area is getting that's being infected is higher and higher and higher and higher and higher, rather than just this is how much it is on one day, then this, then this. The idea is that it's building up, it's cumulative. Because if you had different days, maybe the level of how much that actually looks infected could be different. What they're doing is they're adding on every time how infected it looks. That gives you an idea of how it's progressing. So in this case, the reason is because it allows you to look at the infection over time or the cumulative effect. So it can show cumulative infection over time because it could look, it could appear less infected or more at different time, at different time points or days, really. Here, and you could use some figures as well to sort of describe that. So, for example, here, if we took like C is barely changing at all. If we just looked at it one by one, it might look like this, and it might look like this and this. Whereas we're looking at just the sort of collective cumulative effect of it, where we're looking at just the whole area's leaf and how much it's getting infected, not just like one area, because one area might have more than less, if that makes sense. The clones are planted in adjacent fields in order to control the variables such as temperature, speed, and rainfall. What other two abiotic variables were intended to control? So again, we're looking at things that will screw up our dependent, which is all to do with what area looks infected. So it's basically anything that's going to make the plant look or start tweaking or acting funny. So that's pH in the soil. And another thing I guess you could say is humidity. As well as that, you could talk about the actual content of the water content of the soil, and the minerals, or you could talk about light intensity or how much light there is, like the duration. So two species of chimpanzee, the chimpanzee and the bonobo, are the closest living relatives of humans. And here's a diagram showing the current classification of chimpanzees and humans within this family Hominidae. So there's all our species, there's a common ancestor, there's another common ancestor, there's another common ancestor, and there's another common ancestor. The way I like to think of it is here's your father, and then there's your father's, granddad, your granddad, and their mother, and then your dad, your mom, and then their dad, and their mom, and then I don't know, your dad might have like an uncle as well. I'm hoping I haven't drawn some really weird family tree, and then an aunt here, and then there's you, right? Now, you all have, well, okay, not you all, you and your dad, for example, will have this granddad as a common ancestor. You eventually all kind of branched off that geezer almost, in a way, and your mum as well, and his mum. Whereas your mum does not share a common ancestor with your dad, or so I'd hope, because obviously that'd be a bit dodge. You are less commonly related than these three. Your dad, uncle, and aunt are all siblings from the same parents, so they're more closely related, and they have a more recent common ancestor, whereas your most recent common ancestor is either your dad or your mum. That's kind of what's going on here in a similar sense. So the sooner off you branch, as in like the closer you're branching off together, the more related you are. So humans and chimpanzees are currently classified in the same family. Chimpanzees were once classified as separately from humans in the family Pongidae, along with gorillas and orangutans, and here's a human hand and a chimpanzee hand. So Homo sapiens and then P. troglodytes. Describe two differences in the images that could have been used to classify them in separate families. With phylogeny, what we can do is we can do it on based on genetics, but we can also do it on what sort of physical traits there are as well. I mean, if I have a look at the image here, I mean, they first of all have thicker fingers. I'm talking about the chimpanzee, that is. You can say obviously the reverse. Thicker fingers, or you could say they have longer palms, for example. It's just whatever's describing the picture. You need to make a comparative statement. You can't just say big hand or creases. You're talking about comparative physical statements. Now, differences between the nucleotide base sequences can be used to estimate the length of the time since two species diverge from one another. The greater the number of differences, the greater the length of time that has elapsed from the two organisms that were part of the same species. Figure 19.3 shows a line of best fit for the differences in DNA between pairs of primate species plotted against the number of years since the two species diverged from a common ancestor. Fine. So this is the number percentage. Sorry, that are different. So the best differences in DNA between pairs. So they're different between the pairs of primates, right? So if we look at chimpanzee and bonobo, their percentage difference is 1, 2, 3, 4, 5, 6, 7. So 0.2, 1, 2, 3, 0.3% different between a chimpanzee and a bonobo. Whereas a chimpanzee versus a human, there's 1% difference. Fine. Now it says calculate the rate of DNA change using this data in this figure and give your answer to 3 significant figures. So here it kind of gives away as well what to do because it's percentage per million years, but also a rate means how something changes with something else, right? So how the percentage of change of DNA is changing with time rate. Now, if that's the case, you're looking at the change in the percentage divided by the change in the time, right? Which you have a look as your change in your y-axis over your change in your x-axis. When you look at that, that's known as your gradient. So what you do is you work out the gradient of this curve. So curve for this line. So pick 2 points and work on that. So if I just pick 2 nice points that line up, so for example here, and then, come on, it's got to be like another nice one somewhere. That's all right, that'll do, right? Your gradient is equal to y2 minus y1 over x2 minus x1. So our second y coordinate is going to be whatever this line is, which is what, 3.9, minus our first coordinate of 3, divided by our second's x coordinate, which is what, 21, 22, minus the first's, which is what, 17. If we do that, we get 0.18. There you go. The mutation rate in mammals can vary as much as 20% between species. Use figure 19.3 to calculate the time since the phylogeny of humans has diverged from the chimpanzees and the range over which this estimate may vary. So if we have a look here, we can see that humans, see chimpanzee, bonobo, chimpanzee, humans first dropped at around what, like 5, what's that, like 5.05, 5.5? We'll just go 5. So they dropped at 5 million years, right? Now, the thing here is, though, is the questions told you that there's a mutation rate that can vary as much as 20%. The reason that the divergence happened was because their percentages that are different, right? So at the point where humans are different to chimpanzees, there was a 1% variation, well, 1% difference between the two of them. Now, the thing is, though, they told you in the question that mutations can cause them to vary as much as 20%. So it's actually plus or minus 20% of this. So that means you can have 1% plus or minus 0.2. So the range at which these variations occur actually really is 0.8 to 1.2. So what we do is we look for the 0.8 mark here, right? And we look for the 1.2 mark here, which is there. Can you see how that's actually 5 plus or minus 0.2? It could be anywhere here, like, at this time or this time where they diverged. So it's actually going to be, the time since divergence is going to be like, what, 5 million? And it's going to be plus or minus 0.2 million years. Some scientists have suggested that humans and chimpanzees should be reclassified to belong to the same genus. Evaluate this suggestion using evidence from these figures and your own knowledge of scientific basis of classification of organisms. So when we're evaluating what we are looking at, we're looking at supportive statements, so what directly agrees with what they're saying. We're looking at contradicting statements, what directly goes against it, and then issues with any validity. So for example, if I say, okay, 10 people in a room I gave chocolate all shat themselves, therefore 100% of people will be lactose intolerant. There's multiple layers of just what the hell's going on there, because one, it's only 10 people, small sample size. As well as that, I don't know how they were selected. I don't know if they're allergic to chocolate and this and that. Number 3, you can't generalize 10 people's results across like an entire, every human in the world. And as well as that, you also can't really just use chocolate as a surrogate marker for lactose intolerance, because I don't know how much lactose there is in the chocolate. So it's basically things that kind of get in the way of saying how appropriate this study is or this conclusion is. The data in the table, is it actually appropriate to say what they're concluding? And also, can everyone else do it and get the same results? Can it be applied to absolutely everybody in the world? So we have a look then, we're looking for same genus, right? So domain, kingdom, and so forth, right? So desperate King Philip cried out for goodness sake. So desperate, domain, king, kingdom, Philip, phylum, cried, class, out, order, for, family, goodness, genus, sake, species, right? So if we're looking at the same genus, they're both going to be, the same, for example, Homo sapiens, right? But in the question, you can see here, wherever it's gone, this is a different genus. So we need to look at what's going on here. So if we have a look at that first figure then, we're looking at humans and chimpanzees, right? So we can see where's humans, Homo sapiens. Here we are, right? Now remember that this is all to do with like the questions told you that we've got two species of chimpanzee, the chimpanzee and the bonobo, right? So if you have a look here, humans, bonobo, and chimpanzee. Obviously, bonobo and chimpanzee are more closely related here, but you see humans shared a common ancestor with them and came off the same branch. Fine. And as well as that, on this figure as well, humans, time since they first diverged in million years. So how long it's been since, as in they diverged 5 million years ago, they most recently diverged. So supporting evidence immediately is that humans most recently diverged. Plus, they share the same phylogenetic branch, right? However, what's contradicting about it is that if we look at, for example, the chimpanzee versus the bonobo, the chimpanzee and the bonobo first dropped here, and it also says on the figure that they also came out here as well, and they're more recent. So that kind of goes against it because we're not really as recent as these two. So why should we be put in the same genus? So one thing immediately is that there is what is it? There is the chimpanzee bonobo diverged more recently at how many million years is that? Like 1 million years, and branched off more recently in the other figure, right? But as well as that, another thing that's contradicting is that they also have different anatomy. Their build is different. So that's another thing there, is that they have different anatomy. So now we need to think about criticisms in terms of validity and any other points as well to discuss using our knowledge of the scientific thing. So one thing immediately is that we're trying to look at, comparing anatomy and stuff like that, right? In the past, we used to just look at what they looked like and said, oh, did they look similar? But now we're doing more things like, for example, biochemistry and comparing like their, the antibodies they make, the proteins that they make, the DNA sequences and stuff like that, right? So the whole point is that we need to talk about, why there's a bit of an issue here, really. So in that sense, is that all, number one is, initially classification was based on observable characteristics, but now we're dealing, now you've got your biomolecular techniques and all that kind of stuff, and they're more accurate, right? And also as well as that, scientific knowledge develops over time. Now, as well as that, the species that diverge most recently have the most similar base sequences, which can actually be seen here between the chimpanzee and bonobo, because that's the percentage difference between the chimpanzee and bonobo. So it's probably not wise to just kind of do it based on what we're seeing here, really, that's all. So one type of gene is known as a homeobox gene. The base sequence of homeobox genes in humans and chimpanzees are almost identical. What conclusions about the evolutionary relationship between humans and chimpanzees can be drawn from this piece of evidence? Homeobox genes, remember, they are what contribute to your body plan. So what they do is they code for a part of a protein called the homeodomain, which binds to specific sites in DNA and allows them to work as a transcription factor, right? Now, the thing is with these is that they have changed very little. They are highly conserved. So basically, if they're highly conserved, there's not really much you can get out of them. There's little conclusion you can draw because they're highly conserved in the animal kingdom. A lot of things will be similar, but in them, conserved, right, in the animal kingdom. And because these are both animals, they're not going to be massively different. So out we go with that. Figure 2 shows the disaccharide lactose, which is found in milk. Another disaccharide is maltose. Maltose and lactose both contain the same number of atoms of each element, CHNO. What other two structural similarities are there between these two? I mean, one thing off the bat is that they have a 1,4 glycosidic bond here, because that's carbon 1, that's carbon 4. So they both have that immediately. So this is what you see if you should know the structure of maltose as well, and that's what they all have. Because maltose is another example of a molecule that has a 1,4 glycosidic bond. So that immediately is 1. So 1,4 glycosidic bond. Even just saying glycosidic bond in itself is fine. As well as that, other things as well as that they both have 2 hexose sugars in them. Or you could also say that they have 2 CH2OH groups as well, which both of them will do. Complete the table to identify differences between the structures of lactose and maltose. The first one's been done for you. So with lactose, what's different is that if we have a look, you can see that this glycosidic bond is formed above the plane of these glucoses here, right? Whereas an alpha one, so that's a beta one, sorry, and an alpha one would be below. So immediately, one thing I can say is that we've got beta glycosidic bonds in glucose, whereas in maltose it's alpha. As well as that, lactose has got beta glucose in it, whereas maltose has alpha. And then as well as that, in this sense, what else can we see? Can you see how these are flipped? In lactose, the sugars are flipped, whereas in the maltose, the monomers are the same direction, or their orientation. What was I going to say? Oh yeah, here I'm just being lazy. You should obviously write the whole thing. I'm just writing alpha and alpha. Don't do what I did. Like, just general rule of thumb. So one of the monomers of lactose is galactose. The bacterium E. coli usually uses glucose as a respiratory substrate. Under certain circumstances, E. coli can use galactose as a respiratory substrate by breaking down lactose into glucose and galactose, and then using both glucose and galactose as respiratory substrates. Explain how the structure of it allows to be used as a respiratory substrate. So the idea here is that we're going to be using it to break down bonds and stuff like that, right? Which are going to allow it to be, phosphorylated in this and that. So immediately off the bat, one thing we can say is that because it's similar structurally, it must have the similar bonds which, contain energy. So the bonds containing energy can be broken by the same respiratory enzymes. So can be broken by respiratory enzymes because, if you want to do glycolysis on it, for example, you're going to have to be able to have the enzymes to do it. What else does it say about allowing it to be used as a respiratory substrate? In order to do respiration, you've got to have everything dissolved nicely and soluble. Otherwise, how are you going to move it around the cell? So it's soluble, so it can move within a cell. And you should say why, though, because it's saying explain how the structure of it. This is due to its hydrogen bonds, which form due to its OH and H groups bonding with water, right? You can't just say you can't just say like, oh, and bonds can be broken or the bonds contain energy. The idea is that the bonds contain energy because that's its structure. It's allowed to be used as a respiratory substrate to be broken by respiratory enzymes. And in this sense as well, it's soluble, can move around, right? But that doesn't tell me the structure part. That's only the property, which is due to the hydrogen bonds. So it says E. coli usually grows in conditions where the extracellular concentration of lactose is low. Fine. In such conditions, lactose does not easily cross the bacterial cell surface membrane. It suggests and explain why it's unable to do that. So you need to think in your head why. I mean, generally speaking, the reason we have to use facilitated diffusion for like glucose and stuff is that it's too big. So in this case, it must be too big as well. And therefore, it cannot pass through the phospholipid bilayer. There you go. In order for lactose to enter the cytoplasm of a cell, a protein is required. The E. coli living in the digestive system of young mammals is more likely to contain this protein than those living in old mammals. So again, young mammals versus old mammals suggests an explanation. So again, in order to digest lactose, E. coli needs a protein. This lactose digesting protein seems to be higher in the E. coli in young versus old. Lactose, milk. So to me, it's saying that the young ones are having more milk. So young mammal diet includes more milk. So there's more lactose, right? So here, this question is hinting at the fact we need to think about the fact there's more, lactose in the diet, which means we're going to have more of this protein. So protein synthesis involves transcription and translation. So something's screwing around with transcription and translation. Now, in this sense, it must be something to do with an operon, which is where you've got a group of genes that are controlled by the same mechanism and expressed at the same time. These are more common in prokaryotes because they're smaller and simpler in their genomes, the genomes of the prokaryotes, that is. And it's a good way because it means that you can switch things on and off a bit more easily. So the example you guys need to know about is the lac operon, which is a group of 3 genes, the lac Z, lac Y, lac A, right? These are structural genes, and they code for 3 enzymes. It makes a big fatty mRNA, and it's translated. So transcription, translation, right? Now, before here, we have our promoter. A promoter is a sequence right before any of these structural genes where RNA polymerase will bind to and, get the job done. Before the promoter, you've got an operator, which we will get to, and then before that, you've got a reg gene. So a regulatory gene. What this regulatory gene will do is it will code for a repressor if there's no lactose, right? And if there's no lactose, what this does is it prevents the transcription of these. What happens is this repressor protein is constantly made, and it will bind to an area called the operator, which is close to your structural genes as well. The binding of this stops RNA polymerase from binding to this DNA at the promoter region, which means you can't get transcription and translation. But when lactose is present, what lactose will do is it will bind to the repressor like this, and it will cause it to change shape so that it can no longer bind to the operator and is no longer complementary in shape. Therefore, RNA polymerase can bind to the promoter, and then you get your stuff going. So we need to describe that in this context here. So because they have more lactose in their diet, that means it's going to bind, or the lactose is, binds to the regulatory, so the repressor protein, and that changes shape. So your lactase enzymes, including lactose permease, is expressed. As in summary, the lac operon is switched on. So lactose is a reducing sugar. Benedict's reagent can be used to determine the presence of lactose in a solution. A colorimeter can measure the concentration of lactose. The first needs to be calibrated. I describe how a method that can be used to use this, and a colorimeter could be calibrated to measure the concentration of lactose in an unknown sample. So what we need to do is we're going to use this colorimeter, and we're going to try and figure out how to grab concentrations of a mysterious thing. So what we're going to do is we're going to make a calibration curve, which is where you've got a standard of known concentrations of glucose. Sorry, whoops, other side here. Concentrations of glucose that you know, and its absorbances from the colorimeter. So first of all, we need to do is we need to set up and produce solutions of which you know the concentration of. So you would produce serial dilutions of known concentrations of lactose, because we'll know that if it has an absorbance of this or whatever, then we'll know that it's going to be that concentration, right? So you would do that. And then what you would do is you would zero the colorimeter first using a blank cuvette. And then what you would do is you would use the red filter, because that's what we're going to use, because we want all the other sort of the reason is because then what will pass through it is going to be blue light. If you see an object that's green, it's absorbing all light except green, because green is coming back to your eyes, it's being reflected. So that's why we're going to use a red, or you could say orange as well, filter. Then what we do is we measure, the sample, whatever. We measure your absorbances of all of these samples you've just done of the known concentrations, and you make a calibration curve. And then what you do is you test the unknown sample, and then read off your curve to get its concentration. That's it. Done. So figure 21 shows some of the steps involving producing a genetically modified bacteria. So DNA is isolated from a donor organism containing a useful gene. So we've chopped it out. Here we also made a chop here in this plasmid. Then we're sticking it in here, and we're slapping it back into the bacterium. So in this case, what we've got going on is we've been using a bunch of techniques. So we've been using, for example, here to chop it out, we've been using restriction endonuclease. The same one here will be used to break the phosphodiester bonds here. Then it's going to line up nicely here based on complementary base pairing, because it'll make sticky ends. And then DNA ligase will form this phosphodiester bonds to stick it together. Otherwise, it just kind of line up like that. Then, here we go. You've got your thing inside. So it says a gene is cut from the donor organism using a restriction endonuclease or enzyme. You can say either. I say endonuclease because I'm used to that. The something enzyme is the same enzyme we use, because what it will do, the same enzyme is used to cut an open piece of bacterial DNA so that the base sequence at the end of each piece of DNA are complementary. That's why if you use the same one, you'll make complementary sticky ends. You'll have these bits of unbound on either end that are complementary. So you can put this into the next organism and they'll form complementary base pairs if you use the same enzyme there, like that. So then it says, describe the events that are taking place in the step labeled C. So what I like to think of it like almost is you've got your sticky ends, right? Just like almost like in DNA and RNA replication, your free floating nucleotides, or in this case, your strand or fragment that you want to put is going to line up here by complementary base pairing all nicely like that, right? Although not technically, character helps me make sense of this. Then what happens is DNA ligase is to stick together, sticks them together, puts them like this. So in your same sense with the template strand here and then the DNA polymerase strands and nucleotides together, here, this is floating around. This is almost like the quote-unquote template equivalent in this example, and then DNA ligase will stick them together like that, as in actually form those bonds there, the phosphodiester bonds. So the events that are happening here is, number one, is that you're inserting overall your gene or the fragment, whatever you want to call it, into the plasmid. We need to think about how. First of all, what happens is you get complementary base pairs from the sticky ends, between the sticky ends, due to sticky ends forming hydrogen bonds with the plasmid. And then what you would do is say that DNA ligase joins them via forming, I'm going to write PDE for sure, you write the whole thing, obviously, phosphodiester bonds. There you go. The DNA ligase is not forming the hydrogen bonds, it's making the phosphodiester bonds. It's sticking the two strands together. Otherwise, it just kind of like float awkwardly like here, like that almost. It's sticking them actively. So step D results in a transformed bacterium. Many individual bacteria are not transformed successfully during this. Explain how scientists can determine this. So what they need to do is figure out whether this plasmid's been taken up. So what you can do is you can insert a marker gene at the same time as well, which makes them fluoresce. And then what you can do is you can use, like, for example, UV or like radiation, grow them in agar, and then have a look. Alternatively, the marker gene could be antibiotic resistance, and you can grow them and then give them an antibiotic and see which ones stay alive. So the idea is you're using a marker gene. So insert a marker gene for fluorescence. That's the one I'm going to go for. You grow them on agar, use UV light, see which fluoresce, And there you go. Now it says bacteria can be genetically modified to produce human insulin. The process is similar to that. First, instead of isolating DNA that contains the insulin gene, mRNA that codes for insulin is extracted from the DNA in the human pancreas cells. What needs to be done with the mRNA in order for this to do its job? So here we're using DNA fragments. This is mRNA. So we need to make DNA from mRNA. So we need to make complementary DNA from mRNA using reverse transcriptase. There you go. And here it says, some people are concerned about genetic modification. State one valid concern that people have about this, about when you're modifying bacteria. The idea is that you're going to increase antibiotic resistance. Not immunity, antibiotic resistance. Immunity is different. Immunity involves like your white blood cells and all that stuff, and this is one singular cell, that's it. So there's a risk of increased antibiotic resistance, which can cause a lot of problems. Thank you very much. I hope that helped. Please like, share, and subscribe. You know the drill.